Bit about what we do in my lab. So I'm a molecular and cellular biologist. So that's my training, and we study and look at DNA and how the information in DNA can be used. And as you know, all of us are DNA-based organisms. That's including us. That means the molecule in your cell is made of DNA, and that directs all the activities of the cells in your body. And it's the same DNA that's in plants, and so it's no different. It's also the same DNA that's there in the bacteria that sit on your skin, you know. And so I'm not really a plant pathologist, although I'm going to talk about plant pathology and diseases, plant diseases. But what I do is provide somewhat of a different perspective. You know, the perspective you hear from plant disease is that we need to have resistance, and that's how we combat diseases. Or we've got to kill the organism, you know, or kill the plants that are infected by that organism and sterilize the environment, you know, with chemicals. And what I'm more interested in is in the old adage, you know, disease is really the exception rather than the rule, right? And you may have heard that. And the question is, why is that the case? That's the case because plants and their pathogens have evolved over 1 billion years, you know, since we've had photosynthetic organisms on this earth. And they've learned to live with each other, and the plants have developed mechanisms to tolerate them, and the pathogens, which are really a small group of the microbes. You know, you would say, you know, you have billions more microbes than there are stars in the galaxies out there. So there are more microbes than stars. So it's amazing how much they are. So you just think about the genetic potential that they have. They can overcome anything because they have genetic resources that they can share from the environment, and they continuously do that. They share it horizontally. They don't follow the rules of, you know, you're only supposed to mate with your species. They will transfer DNA. And that's the process of transformation. So I'm digressing. But I just wanted to give you a little bit of background. So when I look at these pictures, like you see here, this is Temecula Valley in 1990s. And you see the devastation that this disease, Pierce's disease, has done. And this is an orchard. I just picked this off the internet. So it's affected by crown gall, and you can see the stunted plants because of the growth of the galls choking the nutritional content and the water relations, because the tumors discombobulate the vascular system in the plant. And so, rather than think of resistance, I'm wondering, you know, why is this, in this case, you know, that old adage, the rule has been broken, and now disease is the rule, right? So what makes disease the rule in this case here with Pierce's disease, and in this case with crown gall? And it's a rule because these bacteria have figured out, they have developed a virulence response that takes advantage of the weaknesses in the plant. So even though it has a very good immune system, there's always weaknesses. And we've seen how the COVID virus took care of the weaknesses that we all have in us. It just ravaged through the community because we had no inbuilt immunity. And the spike protein in the COVID virus has very elegant ways in which to disrupt the immune response. The AIDS virus is really another good example where it actually tunes down the immune response. And so that's what you see in most cases. Pathogens are able to create sensitivity or take advantage of the sensitivity. And so when we think of resistance to diseases and pests as being important ways to conquer the disease, I think it's what I would like to submit to you is to think about sensitivity as also being genetically encoded. And that's what's being taken advantage here. And so what my program has been trying to do is take advantage of this and decrease the susceptibility. So now you have resistance. So when you think about grape wines, it's interesting that we have this composite genetic system where we have rootstocks. It's a composite genetic system, and you have these rootstocks grafted to science. And so there are about 26 different rootstock genotypes that you'll use in the industry, in California. But then you graft that onto more than 150 science genotypes that are. We've been working on just some of these few rootstocks here, 1103, 101. And try to see that these are actually resistant to many of the diseases. They are resistant to phylloxera. So a lot of the work on rootstocks came about after the phylloxera epidemic, in Europe that devastated all of the grape wines. So vinifera, it turned out, was very sensitive to phylloxera. But where phylloxera originated in the United States, those native grape wines co-evolved and were completely resistant to phylloxera. And so now you have a different genotype in the rootstock. And so this provides a really good opportunity to engineer or modify the rootstock without affecting the sign. So we call Cabernet Sauvignon wine because we have Cabernet Sauvignon grapes and they are clones. And those Cabernet Sauvignon wines can be produced from any Cabernet Sauvignon, whatever rootstock it's being grafted to. So the implicit understanding there is that there's no genetic elements that are moving from the rootstock into the sign that's going to change the varietal nature of the wine. So this is something that's been established. And it's true that actually there's really no genetic information that can move past the graft union from the rootstock. Plant cells are static, they don't move. In animals, cells move around, our blood cells are moving around. But in plant cells, they are all static, they don't move. The only way they can move out is if they are encapsulated in seed or in pollen. So that's how that it moves out. And so when you do breeding, you're taking pollen and then you're hybridizing it, and you create these varieties that are like us. They have different parents. So that's why we look different to each other, because we are capturing the genetic changes that were there in our ancestors. And so the DNA in our cells, which is the same in every single, all 30 trillion cells that we have in the human body, have the same genetic component. And that's a living fossil that represents the fossil record of your, and that's the case. So the point I just want to make is that this is a well-established thing. And so when we are thinking of bioengineering, we got to think in terms of when we make changes in the rootstock, that those changes can't be transmitted up, and it doesn't change the varietal nature of whatever is grafted on. And so the idea is to bioengineer rootstocks, and that's what my lab is specializing to do. And the idea is that it fits in really well because it can, what we can do is enhance existing rootstocks. Now, if we can take existing rootstocks and capture whatever they have, good good traits in them, we can remove the susceptibility, we can enhance these rootstocks so that they can be more valuable for growing grapes and sort of maintain productivity and quality. And a lot of that is mediated by the rootstock, because the root of the stalk is what provides all the nitrogen, because the nitrogen comes from the soil, all the elements come from the soil. A lot of the microbes also then come in also through the vascular system in the roots. And so the idea would be to overcome, reduce the amount of pesticides, and that's sort of what excites me. Is we are on this pesticide treadmill, and I'd like to see us get off it and have the roots take care of it, so you don't have to spray. So, for example, in the case of Pierce's disease, since it's a vector-transmitted disease, the only way to interrupt the disease is to kill the vector. And so that's area-wide management with chemical pesticides. And if you see what's happening in the General Beale area, a lot of these insects are now become resistant to pesticides. And so now you're going to have a big problem because of that. But if you could take care of the bug itself, increase the immunity of the plant, then the plant will take care of the bug, and whether the insect transmits the disease is of no concern, because you are stopping it at the level of the pathogen. We have nothing to stop the pathogen. All we can do is kill the transmitter of the disease. So that's in general what I mean by this bioengineered rootstock concept. So these would be the features of the bioengineered root. So there's no movement of genetic information into the sign or into the environment from the rootstock. That can only happen if for some reason the rootstock dies. I mean the sign dies and the rootstock becomes a plant, in which case now it's going to, whatever new information you put in that rootstock will get into the environment, because the pollen will contain that genetic information, and the seeds that come out from those rootstock plants will come out. So there's certainly that caveat. Also, you can graft any sign, and you all are familiar with that, and so there's really no need to modify the sign. So you can maintain the varietal genotype in the way it is intended. You can protect the health and nutrition and productivity of the grafted sign because of the rootstock. You can enhance the innate immunity of the root system, and in most cases you will see whenever a disease hits the sign, it goes down, and once it kills the root, that's when the plant dies. So if you look at Pierce's disease, for example, you will see the symptoms first in the leaves, the scorching, leaf edge scorch symptoms that are telltale symptom for Pierce's disease. But those leaves fall off and the plant's fine, But the bacteria go down into the root, and then they cause stress in the root, and that's when the plant dies. So the picture I showed you of the Temecula Valley, you see a lot of these missing plants, and that's because of the vine death that's associated. And that's really catastrophic when you lose root functionality. And so by having rootstocks that can maintain functionality during an infection, because they are recognizing the pathogen and providing a good immune response, then you're having a more healthy plant. So you can inhibit. So there are various ways you can increase, you can decrease susceptibility. You can inhibit pathogen movement, so it doesn't move or stop it. You can inhibit the pathogen growth. You can inhibit the feeding activity of the pathogen. And this is where we found that actually this is a more tractable. You try to starve the pathogen. It's a feast for them when they infect your plant and take all the good stuff from the plant. You can also interfere with the development process in pests, and so that they don't proceed along their development, and that will also reduce their population size. Once you reduce the population size, then that can be managed much easier by the immune response, in the plant. And then finally, I think this notion that you can stack resistance. That means you have resistance already there. Now you're adding to that, so you're stacking it. This is really impossible to do by traditional breeding, because when you do traditional breeding, you're mixing two genotypes and you're mixing lots of genes. Half the genome is mixed. And so to get two genes that way, it becomes infinitesimally small probability of being able to stack genes. And so this technology allows you to do that. Okay. I just wanted to just go over that, how do we decode. In order to do this technology, you have to understand the mode of action of the disease. It's like a doctor. If you don't know what's the cause, then it's not possible to fix it. And so understanding the causality and establishing cause and effect relationship is really important part of this technology. It's not necessary in reading, because reading, it's a crapshoot. You're just looking at a phenotype, a visual, a marker that allows you to say, well, I've got the trait here. But here you've got to understand really how it works. And this means decoding genetic information. So as I mentioned, most plants and other living organisms have really three types of molecules that perform all the functions in a cell. So you have DNA that, as I mentioned to you, is the same in plants and animals and bacteria and humans. Encodes all the instructions, that's the living fossil. And that's what passes on genetically. So when you have kids, your DNA is being preserved, or at least half of it, is being preserved in your kids. mRNA, this is the decoded information from the genome. So the genome has a lot of DNA in it. Part of it are genes, which perform functions. And those usually are represented as messenger RNA. So when they give you if you had the Pfizer vaccine, that's an mRNA vaccine. What they injected into your body was a messenger RNA. It was coated in a lipid molecule so it can enter your cells. But what it does, it was able to be translated. It gets translated in your body to make protein, makes the spike protein. And that spike protein triggers and educates your immune response that this is the bad guy. We need to have antibodies that destroy this. And by destroying the spike protein, it stops the virus from multiplying. So messenger RNA is really the key piece of information that tells you the causal genes that are necessary. That messenger RNA is made into the third molecule, which is the proteins, and the proteins perform the function. So in the case of the crown gall, I'm going to explain to you how we use RNA. We try to stop the RNA that is responsible for making the tumor through epigenetics. So you have two things. You have genetics that's associated with the DNA, as I explained, because that's the inherited component. But the expression part, when the genes are expressed, that's really epigenetic. That means that's what's regulated epigenetically. You may hear this term more and more about epigenetics, how epigenetics modifies our genome is still, a lot of people are investigating that. A lot of bad habits, for example, are epigenetic modifications that control the regulation of many functions. Depression, anxiety, these are all epigenetic phenomenon that control. So that there are other controls on genes that are not associated with just the DNA sequence itself. And there are processes, for example, the defense against viruses in plants and in humans goes through what is called the RNA interference mechanism. And so that's the mechanism that we've used. It's an epigenetic mechanism that we use to control crown gall. And I'll go into the details of that so you have a better understanding how epigenetics work. But I just wanted to educate you on what is epigenetics and what is genetics. So the way to simply distinguish them is genetics is changes in the sequence of that DNA, that's genetics. But when you have changes in expression that are not associated with changes in the DNA sequences, then we call them epigenetic. It's something other than the DNA that's regulating the expression of genes. And more we find lots of things are epigenetically regulated in humans and in plants. The other area that I'm going to touch on is we've been able to insert proteins into which we call cap proteins that disrupt and degrade the bacterial surface of Xylella. This is the causative agent of Pierce's disease. And by disrupting the surface, we release molecules that educate the immune response that, oh, this is a pathogen, this is not a good guy, this is a bad guy, it's a pathogen. And the immune system is looking at that. It's just like our immune system distinguishes between self and foreign. This is a really important part. How does your immune system look at a protein and say, oh, this is one of the proteins being produced in the body, but this is a foreign protein. It's coming from a piece of the pathogen. So they look at debris, break down products, and they look at that, and that's how you educate the immune response. So the immune response needs an education process. And so by disrupting the surface, we don't kill the pathogen, we just disrupt its surface. It teaches the immune response to, oh, it needs to be turned on. This is why autoimmunity is a really big problem. You've heard of that. There's a lot of diseases in humans, autoimmunity, where tissue rejection. These are all where the immune response turns on yourself, and there are lots of diseases associated with that. So I just want to tell you about that. There's a way to educate the immune response by giving it something very specific. So the next piece that you need to have is that you need to get the nursery into the lab. And so this is technology that we've developed. As you know, all the grapevines, whether they are rootstocks or whether they are scion, they are vegetatively propagated. They are all clones. And that's why they have names. Cabernet Sauvignon, Chardonnay. That means these are clones. Fuji apple is a clone. That means all Fuji apples came from this one tree in Morioka, Japan. I visited Morioka and I've seen this tree, the original Fuji tree. It's still there. It's a relic. It's more than 80 years old. It's sitting there, and all the Fuji apples that we eat came from that one tree. So that's what we mean by micropropagation. Micropropagation is clonal propagation. That means they are copies, they are clones, they are identical. Now there are changes that take place, and that's why you have variations. In Chardonnay, you may have a different Chardonnay clone. That's because they've had single changes in the DNA that give it some unique character, but those changes are not like you would have through breeding, where you're changing half the genome, right? Like it's changing us. I do not resemble my parents, although I contain half of my DNA from my mom and half my DNA. So my DNA has two parts to it, a mom part and a dad part. And in clones, they are just small changes that are taking place. And that's what the technology we are developing can make, those small changes. Induce a single gene or turn off something, That's the approach we are talking about. Okay, so the way we do this, and this is something that we developed, is we take anthers. So this is from the field. These are the male part of the grapevine flower. And it's the anther filaments, this little stalk on which the anther sits, is we put those into culture, and we get what is called an embryogenic callus. It looks like applesauce. Applesauce is individual cells, actually. So when you have a mealy apple, your teeth are going in between cells. A crisp apple, the contact points between cells are highly tight. So when your teeth crunch into apples and you have that crunching sound, you're actually breaking cells as your teeth goes through. And so So these calli, which are unorganized growth, are just individual cells growing. So there are probably hundreds of thousands of cells from that particular rootstock. But embedded in these cells are stem cells. Stem cells, as you know, are those magical cells that have the ability to become any cell type. It's something that we are working, in the Human Genome Project they're working on. They're called pluripotent stem cells. So pluripotent stem cells are those cells that have the capacity to become anything. So in this case, the stem cells have the capacity to become an embryo. An embryo is nothing but a seed. A seed has a shoot part and a root part, and a seed can be germinated. So we can convert these cells into embryos. So an individual cell, let's think of it, can become an embryo. And then these embryos multiply. You can see there are tens to hundreds of embryos here multiplying. Imagine how much space you need to have 100 plants. We have that in a small tube. You can have multiple plants. So imagine if you put new genetic information or modified genetic information in one of those cells, that cell then becomes an embryo, and you're propagating now this embryo as a way of propagating the plant, and then you can germinate them, take them out of the greenhouse, and then you're out into the field. So this is the process. So you have to get them into the lab. You've got to create, if you want to modify an existing rootstock, that means we've got to start with cloning that rootstock into this culture. We create this culture, and now this culture is quite stable. It stays for, we've had walnut cell cultures for almost 2 decades, and they're quite long. And so they have a way of preserving themselves, because that's a natural way in which plants are able to preserve themselves from their seeds, and the embryos that are there in seed. Yeah. Seeds all have embryos, so it's a way of micro-propagating grapevine seeds that are clonal. Just think of it that. So then you need to have a way of introducing DNA introducing new genetic information into those cells. So you need to have some sort of a delivery system. So that delivery system comes from nature, and that's Agrobacterium tumefaciens. So it's the same crown gall forming bacteria. The way it forms the gall is because it has this, it's a vector that carries this plasmid, and this little blue piece here, this DNA is able to transfer. So it's able to infect those plant cells, so it can infect that callus that I showed you, and it'll insert DNA, because that's how it passes this DNA from one bacteria to another. And what we do is we trick it, we sort of engineer this piece and put the genes of interest in that, and it transmits those into the plant cell. Now when it goes into the plant cell, that piece of DNA gets randomly inserted into the genome in different parts. So different cells have the piece of DNA inserted in a particular part of the genome. Other cells will have another part. And so we are able to create events and get plants from those events, and then look at those plants and see which one suits the job, and then we move those forward. So once you get those, you can encapsulate those cells that are transferred as embryos, and then now you singulate the events, and now you have plants that represent those particular events. And so as a consequence, what happens is that you have this new genetic information in every single cell of that plant, because that single cell became an embryo, and all the cells have the new genetic information in a particular location. And when you have a plant, you have the same information in every single cell. So that's important, because you can't have the new information in only some parts of the cells and not everywhere. It's not going to work. So when you change something genetically, you want to change it here. Yeah.
So your cell culture is just from the male part of the plant? So
Yes.
that's only half the DNA?
So it's a clone.
But isn't it just half the genes you need to make a plant?
Yeah, it's a good question. So when you get the half of the gene is when you get pollen. So the gametes have half the gene. So when we look at the rest of the plant, it's diploid. It has both copies of whatever was there in that plant. So it's really a cloning process. When you create the sperm and egg, the sperm and egg have half the genome. So all of us are diploid. That means we have 2 copies of the genome. And in this case, when we take this little stem, we are not taking the pollen. We are dealing with the diploid part. It's a really good question. Yeah, feel free to ask me questions in case.
How are you screening here? By sequencing?
Oh, so the way we screen here, we add a gene in there to give antibiotic resistance, so we can select. So we do add some other genes. So we have the active genes and we have the inert genes. Sometimes we will put it that it gives a certain color, so we can recognize those cells that are transformed from those that are not. So we need to do that to make sure that we have some sort of selection process, because we don't know how frequency these events are. If it's 1%, it's typically between 1% to 0.1%. So of the cells that you have get transformed. So it's a really good question. Yeah.
So why not use phage instead of agrobacterium?
Yeah. So you could use phage. The only thing is viruses. You'd have to use a virus from plants, and they are infectious agents, and so it's problematic. Also, the payload that is there in the virus is small, because many times the virus has a head structure, and you're constrained by how much you can pack into that head, and so the payload becomes a problem. In the case of agrobacterium, the payload is fairly large, so it's like a B-52. Big payload, yeah. So here what we do is we are putting transgenes. In this case, it's the trigger for turning on the RNAi mechanism. Okay, so I'm going to, this is the slow part of it. Now it's going to go a little bit faster. I just wanted to make sure that you all understood the technology. So I'm going to go through the 2 projects that we have ongoing. One with the Rootstock Commission, where we are trying to engineer rootstocks that are resistant to crown gall. So we are working with these rootstocks. Initially, we were just going to work with Thompson seedless as a rootstock, but then the industry said, no, you need to work with some interesting rootstock that we find particularly relevant for the industry. And so this was the choice of rootstocks that came to me, that the commission felt were important. And so we've been working on these. So we created those embryo cultures that I've talked to you. We harvested the filaments and then created the embryo cultures. And then we've developed the transformation system and everything. And what we found was that all rootstocks are very sensitive to crown gall. GRN1 in particular is much more sensitive. But they're all sensitive, so they all make galls, and we can do them in the lab, on the plants in the lab. We can infect them with wild-type agrobacterium. So it's, I'm probably preaching to the choir here. There's, crown gall is one of the most economically destructive diseases. It's been a bane for grapevines since all over the world. All of these rootstocks are sensitive. And the disease can manifest itself in various forms, partly because of how the disease works. It can cause necrosis in the roots, and it's really a bad one to have. So if you can remove that as a possibility by having a rootstock that's immune to crown gall formation. So we focused on the formation of the gall itself, rather than on the bacteria. So So that's again the strategy that we use. And I'm going to explain to you that strategy, why we focused on the tumor. So the tumor formation is the disease. That's the way to think about it. Because if you did not have the disease, you don't have the tumor formation. And the ability to form tumors is the most relevant part for the pathogen. And if you look at all the pathogen and how it's evolving in vineyards and orchards, it's evolving by passing this tumor-forming genes. And that's how it's able to infect. It's able to infect almost all plants. But when you look at the epidemiology of this bacteria, it's now spread all over. There's probably not a handful of soil that doesn't contain this bacteria. It's very well spread out. Recent estimates where they've looked at agrobacterium from 6 different continents, and you see that the same features are there so they've essentially spread, and they spread because of the way we do agriculture, because we are cutting and chopping, grafting and doing all sorts of wounding, and this bacteria really likes wounds. Okay, so what it does is it has 3 transgenes. That means genes that it transfers into the plant. 2 of them actually convert tryptophan into, tryptophan is an amino acid. It's really important amino acid for us. It makes it into indolactic acid. Indolactic acid is a auxin. So it is able to take tryptophan, which is you found in all plant cells, and make it into an auxin. So auxins are really powerful growth regulators in plants. And then it has another gene which is called IPT that works on different pathways. And I know you've got to be a biochemist to understand these, but just to think, it makes a cytokinin. So cytokinins are shoot-stimulating hormones. They also prevent aging in plants. Auxins are used for rooting. So those of you who have used root tone, you dip the edge of your bud stick, in root tone, and then you'll get roots. And that's because of the action of auxin. And it's this amount of synthesis of these 2 hormones that leads to the tumor formation. And the tumor formation is taking place because these genes that they've sent into the plant make messenger RNA that encode these enzymes that then, so those are translated into protein, and these enzymes turn into a factory of making auxin and cytokinin. To make so much auxin and cytokinin, that's probably the reason why the roots die and you have aging process related to crown gall is due to that. So our strategy was to turn on the immune response that is already in plants. This RNAi mechanism, as I mentioned, is a defense response against viruses. Because what viruses are RNA, most viruses are RNA, and in plants, most viruses are RNA. So they are really infectious messenger RNA. And so the way the immune response works, it looks at double-stranded regions of the RNA. And so if you make a trigger, so what we've done is created a transgene that's a trigger to allow the immune response to recognize this messenger RNA as foreign, and it knocks it out. So we've created these, so our tDNA that we've sent in has 2 trigger genes. That means they make double-stranded RNA. So you can see this is a double-stranded RNA against the IPT gene, and it's one of those IAM genes. And once we make that double-stranded RNA, it wakes up the immune response that's already there and says, oh. And what that does is there's an enzyme called dicer. It takes these and dices them up into 20 nucleotide pieces. And then those are taken up by another enzyme. This is already part of the immune response. This immune response is there in humans, plants, and most eukaryotes have this, what is called RNAi. This is what I call an epigenetic response. The RISC complex uses these small interfering RNA as a guide. And so it'll take out just that messenger RNA. Imagine, if you're going to destroy a messenger RNA, you're going to disrupt everything in the cell. But nature has provided this very unique, very specialized. It's like a rifle shot. A sharpshooter will just take out that one it just takes out that one messenger RNA, nothing else. All the other messenger RNAs are fine. It's because of these chopped up pieces, which are 20 nucleotide long, and the probability of matching perfectly 20 nucleotide long piece is infinitesimally small, so you can knock out one gene. So the moment you go, the probability of 16 bases and above means you're only looking for the one gene. So it's just the probability of having the sequence repeat itself. And so it's a very intelligent system. This RNAi system, it's already there in the plants. All we are doing is alerting it, hey, how about destroying these couple of messenger RNAs? And when we do that, completely, you see no galls. You can infect them, the galls are gone. The bacteria are fine. The bacteria don't move. And one of the things we see is that you can see here. This is a tomato which we have engineered. You can see this is the wound site, and you don't see any cell division taking place. This is wild type, and you can see all the cell division taking place. This dense small cells. One of the interesting things we learned is that if we graft, so this is a resistant, crown gall suppressing rootstock to a wild type scion, and then we cut through the graft union and put Agrobacterium tumefaciens, and you see that the galls are formed in the scion, but the moment you hit past the graft union, you get no gall. So that means these small RNAs don't move in the cell, so they stay in the graft union. So that means it'll be able to protect the graft union from crown gall. It's not going to protect the top of the part of the plant in case the bacteria goes. So that's something to understand. It also doesn't kill the bacteria, right? Because you're only stopping the gall, you have nothing to, the bacteria can still keep infecting, but every time it infects, it doesn't have a gall, this hotel to feed it opines, which is what the reason why they make the galls. They make these nitrogenous compounds by parasitizing the plant metabolism. And the opines can only be metabolized by the agrobacterium. So that's one of the reasons. So we've made some progress. We've now have some lines that are sort of for each of these rootstocks. We now have lines that show no resistance. So you can see here these leaves, we can form galls on the leaves, but these are the resistant the plants that suppress gall formation, and you don't see any gall formation. So the next thing would be to sort of take them to the greenhouse and then get them out into the field. Now I'm going to talk a little bit about how we are dealing with enhancing the rootstocks against Pierce's disease. So you have some idea of what we're doing there. So here, in the case of Pierce's disease, it's a transgraft protection. So in the previous one, I mentioned to you that it's only graft limited. That means everything below the graft is protected because the small RNA doesn't move. But we've discovered long ago that actually you can move things from the rootstock to the top, and that is proteins. So if you secrete proteins into the apoplast of plants, they will just move past the graft union. And so we came up with the idea then, how about having a rootstock that protects. And so this is where we are moving these cap proteins. All these cap proteins, the way they work is they disrupt the surface. So they are made by the rootstock. So we have genes that encode these cap proteins, and they're secreted in the apoplast, and then with the transpiration stream they go up the plant. And then, so this is the ones, they disrupt the surface of the plant. So we have really transformed just two rootstocks, 1103P and 101-14, and we've grafted them with Chardonnay scion just to do the test. So we have many lines, and we are testing them out in the field at the moment. in order to do this test, we had to go through and get a permit to do the testing. So they are under a federal permit, and the first planting was made in 2018, and 2019 we made the next planting. And so we have them, the rows go north to south, but the cordons, we have an east cordon and a west cordon. The east cordon is the one where we've been doing the inoculation. So we're doing inoculations on shoots that come off the east cordon. And we've been doing these, so we infect them. So we are the sharpshooter. We put the bacteria, about 20 million bacteria in each inoculation on the shoots. And then on the west side, we don't inoculate, so that we are just looking and we compare the yield on the east side to the yield on the west side. And then when the season ends, then we look and see those shoots that were infected, whether they show any symptoms. And one of the key symptoms is bud break. The bud break is really inhibited when it's infected with Xylella. So you can see these plants. You can see the yield here. They are all yielding really well, even though they've been infected with Xylella for at least 3 years now. So we're going to do a 4th infection this year. They have all show really good bud break, so they don't seem to be suffering from the disease, in spite of the fact that here we have the rootstocks sending these proteins up, right? And we are able to protect the plant. And the symptoms are, in most cases, less, than what you'd expect. So we have we are seeing these many lines that are showing some promise, and so, something we would like to move forward with. So I want to talk a little bit about the federal regulations, because I think that is something that may be of interest to you, or maybe you all don't know about these federal regulations. So there's a coordinated framework of federal regulations, okay, that's mediated through APHIS, EPA, and FDA. Well, APHIS is really USDA. APHIS, the Animal Plant Health Inspection Service, is part of USDA. And so what they do is they use existing statutes to cast regulations. So the regulations that we have are based on existing statutes. So USDA has what is called PPA, the Plant Protection Act, and the regulation that affects these type of plants is what is known as 7 CFR 340. That's the regulation that's used to regulate bioengineered crops. Okay, those that have new genetic information that's introduced into them, right? And I'll explain how that works. Then EPA is much more. EPA is the 800-pound gorilla of regulation. We are just beginning to interact with them. So they have TASCA, and FIFRA is really where they regulate engineered crops. And they regulate them on this notion that they are plant-incorporated pesticide. So let's think about it. They are saying that the crown gall plant is a plant-incorporated pesticide. It's not really a pesticide, right? It's an RNA. RNA has GRAS status. Everything you eat has RNA in it, including the viruses that infect you. And so there needs to be an education process with EPA to understand that this is not a pesticide. We are controlling the growth of the tumor. We are not killing the bacteria, as I've explained to you. So this is where I think your interaction would be really good to tell them, hey, what's going on? And then you have FDA, that's the Food, Drug, and Cosmetic Act. And they are really looking at food additives. So now if you have this, now it's in the food is an additive. But the beauty of the rootstock is there's nothing in the top that FDA is. But FDA has to rule that there's no additives in the top, or there's no change in composition in the top. From a food safety side, and from a food safety, is it an allergen? Is it a toxin? That's something they're doing. So that's how the regulation works. These regulations, just gives you a little bit of history. In '86, they established this formal policy of 7 CFTRI Part 340. That's the really key one that USDA is the lead agency, so they are the ones who regulate that. So they determine the regulation, and they are the ones who coordinate the regulation with the other agencies. So it starts with USDA, and then USDA informs EPA, and then who informs FDA. They began modernizing this. The modernization process of this particular regulation began in 2015, and in 2021 it's completely modernized, and so it's really different. Previously, the way they would regulate is that if you had a sequence from a pest, now the IM and IPT genes they come from the bacteria. Of course, we have made them in a way that they'll form a hairpin and a double strand, but the sequences come from a bacteria. So because they come, so you're guilty before you can prove your innocence. That's how the old system worked. That means you are propagating pests by having the sequences of a pest in the plant, right? Now the modernized version is, okay, we need to make the determination. You tell us what's in the plant, and so you're innocent, and we got to prove you guilty. So that's how the new regulation works. It's much more easier to work with. So I think going through APHIS is going to be much easier. So this is how the modernized versions of these regulations work. Plants with modifications that could not have otherwise been produced through conventional breeding. So if you can prove that the changes you have made are no different that you could make from breeding, no regulation. So genome editing, where you are going in and changing, and I've not talked about genome editing, I've just talked about transgenesis. Likely you can make the case that it's free. However, if you have plants, where you introduce a gene, and then what happens is they have to determine whether there is a plausible pathway for increased pest risk. Until USDA determines that they're unlikely to pose a pest risk, they will not let it go through. So they make that determination by looking at the sequences and trying to understand. The pest risk is based on the plant, the trait, and the mechanism of action. So here, this is where mechanism of action becomes a really key player. If you don't understand the mechanism of action, you really can't get it through the regulatory process. And it's not based on the technique used to produce the plant. This is the same problem we've run into with the labeling. We do not label the process, we label the product. That's how labeling works. So, but it's the same notion. You can do whatever you want. You can use transformation, you can use genome editing. They're not bothered about it. They are bothered about what's in the plant, what's the mechanism. And if you fulfill those, and we've been able to do that, so if a plant does not, and even if you don't meet this exemption, you can go back to them and they will relook at it. So the key thing is the biological properties of the plant, the trait, the mechanism of action. Okay. So this is the process now. What you have to do. It's called the regulatory status review (RSR) process. It takes about It should take about 5 months, but it takes about a year. And then you go through EPA. EPA looks at the molecular analysis. They want to see the products of the genes you put in. They want to establish tolerance limits, how much you have, what are the active ingredients, what are the inert ingredients. They're treating it like a pesticide. They want to know whether it has any allergen, it's an allergen, or it's a toxicant. And the way they determine that, they just run the sequence of the gene through a database and see whether it's similar to anything. It's not very difficult. But it's just a lot of paperwork, and the process is very slow. And for FDA, it's a composition analysis, and if you can prove that it has GRAS status, generally regarded as safe, RNA, we've been eating RNA for a long time. So that's the regulations. Also, IR4 can be involved in helping with regulation as they do for many of the orphan crops. And so this is a good way to engage IR4, and we've engaged IR4 to help us. So this give you some idea. This is what we've done. We've done this in walnuts, and you can see these are the wild type walnuts having crown gall, and our transgenic one has not showing any gall. We started the process, and just in September last year, we received this letter from APHIS saying that it's free, and you can go and plant these. You don't need a permit to plant these. And I think that's going to be important. If we can get it through APHIS, then at least y'all can start testing it and seeing whether it's useful, in the field. And then go through EPA and FDA. This shows you that the small RNA, molecules, you see a lot of them in the rootstock and at the graft union, but the moment you go above that, you don't see anything. These are for the active ingredients. Inert ingredients, you don't even find them. They're very hard to see in the plant. Okay, so I'm just going to, moving forward, so there are two really bioengineered rootstock strategies. One is the graft limited one, that means it stays in the rootstock, and then the transgraft one, which means it can go past the graft union. And that's how you can protect cyan. Now, not all diseases can be protected. You would have to find a way to deal with it. This is the label. It's called a bioengineered label that will be there on these plants, but that label will only be there in the nursery, because the nursery needs the label to distinguish between the engineered rootstocks and the non-engineered. Beyond that, there's no labeling. And even the labeling, if you see the new labels on the Arctic apple and all, they are just many times just stickers or something that you can, or a barcode that you've got to read and go to a website. So I don't think labeling is going to be an issue. I think that's one of the questions we're asking. And this is just a summary, what I mentioned to you, summary of the bioengineered rootstock. There's no movement of genetic information. I think this is the key. The sign can be a non-GMO. And it will be a non-GMO because there's no way you can trace anything. There's no movement of genetic information. You can do all the testing you want in the world. You're not going to see anything in the sign, even though the rootstock is engineered. The label will be at the nursery, but usually not beyond that currently, based on the current laws. It protects the health and productivity of the grafted science, enhances the innate immunity. We can stack. We can eliminate pesticide usage, which is something I've been very much interested in. That's why I feel these are more organic than organic. If you look at all the things that you have got to do to spray on organic, and we were talking before, copper is one of them. It's amazing how much copper sulfate and copper products that we spray against bacterial diseases. They are so profound.
Can you use these and still get organic certification?
I don't know. It's a good question. I think it'll be worth petitioning that and showing that you could. Because I feel you're having the plant take care of itself.
I think CCOF says no. Well, there are no GMOs, but I know at least ROC says no. They've named interface or interference, and they've named some of it, but I don't know if the USDA CCOF. I don't know if they have. I mean, it's mentioned in some of the ROC things, if I remember, but I don't know if it is.
Yeah, I think this is something the community we need to work on and educate the.
That's the case for the graft for the crown gall, but how about the PD example?
Yes, the same thing. There's no movement of genetic information, so
it's just a protein.
It's a protein, so you'd have to, and it's not something you test for. It's going to be, we don't know how much protein will be there, and it's going to be, and there are lots of proteins that move from the rootstock to the scion. So as long as I think the protein doesn't have any allergenicity or toxicity, I think it would then become an issue, because EPA and FDA look at products of the genes that you put. APHIS only looks at the gene sequences. They're not interested in the products of the genes. And so it's a relevant question for...
Could you say a little bit more about the PD resistance that you're doing? What you're actually doing, what you're transporting? Is it something to kill the Xylella, or?
Yes, so it's something that disrupts the surface of Xylella.
Okay.
So it's got protease-like activity, and the proteases, and you find that in xylem fluid, there's lots of these hydrolytic enzymes. So most pathogens use hydrolytic enzymes, that they break down pectin, or they break down different parts of the plant cell wall. And the plant is trying to degrade them. For example, our neutrophils in our body, they are the ones that defend against bacterial infections when you have a cut. So the neutrophils will swim to that spot, and they release these granules. They're called granulocytes, so they have these granules. And these granules have an enzyme called human neutrophil elastase, which is a protease. And all it does is break down. It also makes bleach, a version of bleach, a very oxidatively strong oxidant that kills the bacteria. So it's a normal way in which the immune response is working, and we thought we would include these hydrolytic enzymes. So that's what they are. They are the one part is, we call it CAP because it's a chimeric antimicrobial protein. That's two activities. One, it disrupts the protein, and the other one disrupts the membrane surface of the bacteria, because the bacteria are able to protect themselves. They have like a cloaking device, like the Romulans had this cloaking device. So you don't see, the immune system is blind because they've got this polysaccharide layer on the top that it can't get to, because if you can go through that, so that's where the lytic peptides work, because they are like a knife. They can go through a layer of that and start degrading, and then the protease degrades. Now you release the pieces that trigger the immune response. So the immune response is, it's called they have these they're called DAMPs, or damage-associated molecular patterns. And so they look at these damaged pieces and they recognize, oh, this is from a virus, and then the immune response. In our case, we generate antibodies. In this case, it works a little bit differently.
Is it Xylella specific, or is it general bacterial?
It is not Xylella specific. Yeah, so it could. So a good question would be, how does it affect the microbiome of the plant in any way? Yeah.
Well, I'm just curious, because I don't think once it was created, nobody bothered to figure out how it actually worked. The question is like O3916. The reason we use O3916 rootstock is for the family virus. I mean, yes, it has nematode tolerance, but really it's the virus that is the problem. The O3916 doesn't actually confer true resistance. I have O3916 that will show symptoms of family virus. It's just so mild that it doesn't do anything, but you can find it. You can find the yellow banding a little bit on some of the basal leaves. So the question is potentially we already have something like this that actually already exists as a product that's been around for years, because my guess is they made that at Davis, and they said, hey, it works. They never figured out or bothered to figure out why.
Yeah.
So how it works, because that's expensive. And
that's the expensive part of the research, is really figuring out the cause and effect relationship. And that's really important when you want to tune something up or tune something down. You really need to understand what, so that's what we spend a lot of time on, is on the design part of the strategy, is to figuring out the mode of action. And you see from a regulatory standpoint, that's all they're interested in. Because if they don't understand that, they can't recognize where the safety issues are. And so we've been living in an environment where we have plants come through breeding, and they're just Okay, that's fine, but, yeah.
Any more questions?
Are there any other diseases you're looking at for similar mechanism?
Yeah, so we are trying to develop the similar RNAi technology against Phytophthora, and also against nematodes. So it turns out that if...
What about oak root fungus?
Pardon?
Oak root
Oh, possibly. Does it invade the plant? It invades plant cells. So what happens in many of these cases, they will have a feeding structure. So we still don't know how the RNA, when the RNAi can go through that feeding structure, but it looks like it can go through. So it's possible that an RNAi strategy could work against any of these. And the RNAi strategy is very powerful because, plants already have all the components to set it up. All you got to do is provide the trigger. And once you give the trigger, it turns on the immune response. So it's possible, and that's something that can be stacked, right? All you're doing is trying to put another trigger, So that's what we think, at least on the RNAi side, that's easily stackable technology. Yeah.
Is there a sort of top end limit with stacking that you would stack and stack and stack and you don't get any better, or does it always get better when you add more of the genetic component?
So the way to think of it is there's really no limit. It's the targets are independent. So the way these technologies working, each trigger is a different target. It's sort of like an MIRV, right? You launch this, and there are multiple things going to different locations.
It's like we're getting multiple vaccines.
Yeah, exactly, yeah. There's no interference. I don't know whether there's a limit on the system, whether it would. It's a very robust system because, it's the defense response against viruses. It's what recognizes the multiplication of viruses inside your cell. That's how this system works. And it's deployed for all other things. So it can be autoimmune style where it turns off, they've been able to see in Icelandic populations, they can see a series of times when they've had feasts and famines over the decades. You see the suppression of different aspects of the genes in your body. So the epigenetics area is still a work in progress and understanding how epigenetics works. But this is one part of the epigenetic landscape. We know a lot about the genetics, and not all of it. It's relevant to cancer and so on and so forth. But the epigenetics is a whole new area, that how the brain functions, epigenetics is involved in that, habits, and can you reverse them? So this is also going to be. Now, a good question is, can this system be suppressed? And since it's a defense response against viruses, viruses have developed suppressors of gene silencing. So many viruses have that trick up their sleeve because it's an evolution process. So you expect, and so now we've got to find a way to counteract that. So it's going to be a continued work in progress, I think.
Well, it also sounds similar to the way in which in the 90s they fixed papaya green spot.
Yes, exactly. Now it turned out what they thought they were doing, because they were expressing the coat protein, and they thought, if we express the coat protein, we can encapsulate the RNA, and then won't multiply. But it turned out that the ones that were very resistant didn't make any coat protein. So the mechanism by which it was working was the RNAi mechanism.
That's what I thought.
Yeah, it was the RNAi mechanism. Very stable. It's impossible to beat because when you think of antibody-antigen interactions, they are hydrogen bond interaction. There's a contact points are a few. So if you change those contact points, you change the ballgame. That's why, the COVID virus has had so many versions of it. All it does is it makes mutations, and now it can slip away from the grip of the antibody. The antibody can't be held tight. With RNA, you have a huge long sequence. You're never going to ever get past that. So you're not going to get resistance to RNAi. Because just of the sequence, the probabilities of changing. If you change that sequence so much, it defeats the gene won't work itself. In which case, that's what you're seeing with the COVID virus. It's able to make the small changes, but then it becomes less infectious, or it becomes more infectious. You see some differences. So that's the way to think about an RNAi strategy as being much more robust.
Great. I think that's all I have.
Okay.
Any other last questions?
Oh, thank you.
Interesting.